Hi friends, my name is Janvi Doshi and today we will be studying about useful and harmful microbes. This is a chapter of Standard 9th textbook and this I am doing as a part of my community service. Now what are microbes? Microbes are organisms that we cannot see with the naked or bare eyes. That means we can't just see them, we need a special instrument to see them. Now, what's that special instrument? They can only be seen under a microscope, and that's how a compound microscope looks like. Now, what there are different types of shapes of microbes. Okay, now the different types of shapes exist for bacteria. First is coccus, cocobacilli, vibrio, bacillus, spirillum, and spirochetes. So these are the different types of bacteria and their shape. So if I have Vibrio cholerae, Vibrio cholerae would look like this in shape, and if I had a Streptococcus, then it will look like a coccus in shape, right? Now, let's come to useful microbes. We've always seen that bacteria is bad, but why is it bad? Right, why is it bad? And if there is the bad bacteria, there are also some of the useful bacteria. Now, what is one of the useful bacteria? It's lactobacillus. Now, these are rod shaped bacteria. These are anaerobic bacteria. Anaerobic bacteria means that these bacteria do not require oxygen to multiply. So they are called as anaerobic bacteria. Right. Then we have uses of lactobacilli. What are the uses of lactobacilli? They're used in all dairy products like buttermilk, yogurt, cheese, butter, etc. Large scale production of pickles, coca, cider is also made by one of the lactobacilli. They also treat abdominal discomfort. You see those Yakult bottles, and you eat dahi when you are sick, when you have abdominal discomfort. So that's the use of lactobacilli bacteria. Leaven fodder is often offered to cattle. Now, what does leaven fodder mean? That means the fodder that they give, the food that they give to the cattle, is not just normal food. They leaven it. Leaven in the sense they make it like a mixture of lactobacillus bacteria and some other nutritious bacteria with the food that they have. They make a churn of it and then they feed it to the cattle so that the cattle always remains healthy. Making of wine. Yeah, wines, curdling process. Now, what is a curdling process? You must have always seen your mother cooking. Now, when you see your mother's cooking in the kitchen, she also sets curd. Now, how is curd set? She'll put just one, two, three drops of curd inside a container of milk and she'll keep it for some time. And after some time, when you open it, you'll realize that the curd has been formed. So what happens in that? Milk contains lactose. What is lactose? Lactose is the milk sugar. Like we have glucose, that way lactose is the milk sugar. This lactose gets converted to lactic acid. Now why does it get converted? Because of fermentation. So what happens when I add those 2 3 drops of curd into the milk? The pH of the milk decreases. Right. When the pH of the milk decreases, coagulation of milk proteins take place. What is coagulation of milk protein? See, as the pH decreases, you see that the water gets separated. The there are some certain blocks that start forming, and these blocks come together, and that is called as coagulation of proteins. And that coagulation of protein results in the completion of the curdling process. Right now, how do we prepare a smear? How do we know that when we want to see under a microscope, how do we know that it's been successful? So, what is the main thing that happens? We first take a swab inoculum. You see this thing, right? So, first, it's the bacteria is mixed with certain chemicals, and this chemical chemical and the bacteria that are mixed, the mixture of that is taken on a swab, and then swab is rubbed onto the surface of a slide. Then this slide will further be mounted, right? Similarly, this is a blood swab. What do they do? They do take a drop of blood. They take the slant slide a little backward, and then they take it and stretch it forward at an angle of 30 to 45 degree, so that you get an even blood smear, so that you can see whatever is there in your blood. So that's how you prepare a smear. Now, how do you observe under microscope? First, you make a smear like we saw in the previous slide. Then we stain the smear. Obviously, we have to stain. Otherwise, what will happen? That suppose if I have white blood cells, I have red blood cells, I have platelets, everything will look same to me under the microscope because I've not stained them. So when I stain them, suppose red blood cells are going to look a little purplish in color, white blood cells are going to look a little whitish purple in color. Only the granules and the nucleus will get stained in the white blood cells. That's how you will come to know that okay, this is a white blood cell. So that's how you differentiate, and that is the use of stains. Now then you observe it under oil immersion lens. So you put a drop of oil on that stain slide. You take the oil immersion lens. What's the oil immersion lens? It's the 100x lens. So you adjust your microscope such that the 100x lens is directly submerged into the oil, and then you kind of try to focus the slide on 100x, right? What's an alternative method of making a smear? So the generally oil immersion lens is used uh, for bacteria that we want to see that we've already killed. Now, for example, I want to see the bacteria moving inside the particular smear. So then I can't use an oil immersion lens. Then I'll have to use a 45x lens, and I will have to focus on that particular thing. Right, so 45x can also be used. So this is how you see a stained bacteria, and this is how you see under a normal 45x microscope. This is stained, it looks quite clearer to me. Over here it's not stained, it looks a little lively to me, it is super crowded, and we can see that this is how the difference will be of the same bacteria that's seen under 45x microscope and under the oil immersion microscope, right? Now, this is cultivation of microbes. Now, there are two ways you can cultivate microbes. First is on a petri dish, second is in a test tube. You see that this test tube is slightly turbid. I can't see our part test tube, okay, right? I cannot see what is behind this test tube because it is a little turbid. And why is this turbidity? Because there are microbes in it, right? So that is how you cultivate in a test tube and in a petri plate. So what they do, the microorganisms that they want to grow, they'll just swab it on a plate. And when they swab it, like we saw making of smears, the same way they do that with the plate, and then they incubate that plate. What is incubation? Keeping it for some time. For some long time they'll keep that plate, and after some time when they remove it, they will come out with so many organisms that have been grown, right? So that is about it. Now let's come to probiotics. What are probiotics? Now, probiotics are useful bacteria that are added to our food so that they protect our body from the attack of harmful microorganisms. So, you see that these two good bacteria are beating this bad bacteria and trying to kill them. That is what a probiotic does. The most important probiotic that we all know is Yakult. These food improve digestion and our immunity. So the better the digestion that takes place in your system, the better you see that these foods improve our immunity as well, right? Now, let's come to Rhizobium, which is also a symbiotic bacteria. Now, this you must have seen that whenever your mom gets a methi at house, you can have a close look at its roots. At its roots, you'll find such nodules. What are nodules? Circular type shapes that you see here, those are nodules. So this are called as rhizobium, and these are called as symbiotic bacteria. Now, these are also called as nitrogen fixing bacteria, and they live in nude nodules and supply nitrates, nitrites, and ammonia to the plant. So, as we all know, we also require certain nutrients to function. Similarly, there are some plants that require nitrates, nitrates, and ammonia to function and grow. And that's why we these root nodules and these bacteria that are rhizobium help the plant to get nitrates, nitrites, and ammonia. Now, rhizobia in exchange get energy from them. So it's a symbiotic relationship. If I give something, I get something. So if rhizobia is giving nitrates, nitrites, and ammonia to the plant, what is rhizobia getting itself? It's getting energy, right? It's getting a place to stay. So it's a good thing, and that's why this is called as symbiosis or a symbiotic relationship. Now, rhizobia produce nitrogenous compounds from atmospheric nitrogen. You know, our atmosphere has nitrogen. Yes, so rhizobia is going to use that atmospheric nitrogen and fix that atmospheric nitrogen in the root nodules of the plant. Right, and that is how it is going to form nitrogenous compounds. Now, leguminous plants have rhizobia in their root nodules. Plants like beans, peas, pulses, all these plants are going to have rhizobia in their plants. And the use of rhizobia has reduced the use of chemical fertilizers to a great extent. And it's really good. Okay, so this is how a typical rhizobia looks like when you crush that bean in on the slide and watch it under microscope after staining. You will see that this is how they look under the microscope. So, how is rhizobia reduce the use of chemical fertilizers? So, fertilizers generally provide nitrates, nitrites, and ammonia to the plants. But just imagine if the plant already is okay with having rhizobia in its roots, that means you won't have to use chemical fertilizers. That means there will be less of pollution. That means there will be less of use of chemicals in the food that we eat, and it will be more of organic, right? So that's why we need rhizobia. What is rhizobial inoculation? That means how am I supposed to use rhizobia as an inoculation in my plant? Or how am I going to inoculate rhizobia into a particular plant and have it make its own nitrogen? So first, seeds and rhizobium solution. There are two. First is the rhizobial solution, then there are seeds. Then seeds are coated with the solution. That means seeds में वो solution डाल देंगे. They will coat that seed with the solution. Yes. Then these coated seeds will be sown. So now the seed is completely covered with the rhizobia and now it's been sown. Sown in the sense they're planting that seed. Now rhizobia enters the plantlets. So as soon as the seed sprouts, as soon as the seed is made, rhizobia will enter them. And when rhizobia enters them, useful. So when rhizobia enters that, the rhizobium bacteria is going to fix itself into the root nodules, and this is how it's going to be useful for the non-leguminous plants. So we know that leguminous plants mein to rhizobia is going to be there for 110%, but if there are certain plants which do not have rhizobia, like non-leguminous plants, so these plants will also be able to have rhizobia, and there will be less use of chemical fertilizers if we use this method, which is rhizobial inoculation, right? So that is that was it. Then the other type of bacteria is yeast. You must have seen that when making cakes or breads, we always see that we use yeast. So this is how active dry yeast looks like. Now yeast is unicellular. It is a eukaryotic cell. Eukaryotic means a single cell. Yeast is reproduced by budding. Yeah, yeast reproduce generally from one organism. It gives rises to another by budding. It is heterotrophic fungal organism. What is heterotrophic? It can't make its own food. It's going to survive on someone else. It grows on carbon compounds. Carbon compounds, like you must have seen yeast growing in bread. You must have seen yeast growing on other carbon compounds. So that is one thing. Then how does yeast look under microscope? So this is how it buds. See, this is one single cell of yeast and there's this small outlet that's coming out. You see, this is one cell, there's another outlet that's coming out. It's almost time that they both separate, right? So that's how they reproduce by budding, and that's how the budding phase looks under a microscope. And the doubling time for yeast is 90 minutes. So suppose for example, agar if I have 100 yeast cells in one particular at this particular time, so after 90 minutes, if I'm going to see them, they're going to double in number. Yes, now alcohol production by yeast. Molasses are fermented with yeast. You know what is molasses? Molasses are like when you go to a sugarcane, like when you go to a person who sells sugarcane juice, he वो जो कचरा रहता है sugarcane का, that कचरा is called as molasses, and that is fermented with yeast. That means yeast is added to there. And when yeast is added, ethanol is something that you get as a primary product, and ester and other alcohols are secondary product. So now they produce this ethanol thing, then they manufacture it, then there are certain chemicals that they form out of it, there are certain alcohol that they form out of the fermentation. So that is what it is used for. Now, ethanol and its uses. What are the uses of ethanol? Spirit and other alcohols are obtained from ethanol. You know, spirit, when you go to hospital or when you go to a doctor and the doctor rubs your hand before giving an injection to you, that particular solution with which he rubs your hand is spirit, right? That's the hospital smell. The hospital smell is generally due to spirit. Ethanol is smokeless and high quality fuel. It is smokeless, so it causes less of pollution. So then your question will be, ma'am, why are we not using that as a, you know, why are we not using that as a fuel? But it is highly inflammable also, so we can't use that. Then maize, barley, and other grains can also be used for production of ethanol. It's not just that molasses, maize, darkness, I'll get ethanol, but if I ferment maize, barley, and other grains, I will also get ethanol by that. Now, glucose and fructose can also be used to produce wine. Glucose and fructose can obviously be used to produce wine because it is also sugar, and sugars are easily fermented, right? What is bioremediation? Now, bioremediation is a biotechnical process, right? It's a waste management technique. It involves the use of organism to remove or utilize the pollutants from the polluted area. You see, this entire thing is polluted, and there are certain microbes that have been introduced in here so that the pollution is reduced. So that is what is bioremediation. That means introduction of useful microorganisms into the environment so that the environment is not harmed, but the environment also gets clean. So that's the use of bioremediation. Now there are a few microorganisms that can be used in bioremediation, and some of them are Yarovia lipolytica. That means it absorbs toxins released while manufacturing of palm oil, heavy metals, and minerals. So it's really important. Saccharomyces cerevisiae. It absorbs arsenic, which is also a very lethal pollutant. Then there is alkani borax. Alkani borax is used for oil spills in oceans. Then we have Pseudomonas, Aeromonas, Moraxella, Flavobacterias. So these degrade major pollutants from the environment when introduced into the environment. Then there is Pseudomonas putida. Now what is Pseudomonas putida? It degrades oil to degradable plastic. So it is converting oil into degradable plastic. So generally used when oil spills take place in oceans and all of that. Yes, so I hope that whatever we've learned about today is clear to all of y'all, and I hope you will like my video, share, and subscribe. Thank you.